First of all, I would like to thank the organizer of this forum dedicated to plant imaging because it's a great idea. It allows to know each other better, and it's a good moment to exchange ideas. So I'm Lisiane Brocard, and I'm a technical manager of the plant imaging facility of the Bordeaux Imaging Center. And before handing over to Clement Chambaud to talk about correlative electron microscopy, I will give you a brief introduction about France BioImaging and about its plant expertise. So France BioImaging is a distributed research infrastructure with 8 regional nodes across France, plus a transversal node, which is dedicated to bioimaging data. And France BioImaging gathers 23 imaging core facilities, which are linked to 68 associated R&D teams. And it represents a technological park of more than 350 setups, both homemade and commercial. And last year, we gave access to around 7,000 users. France BioImaging collaborates with the academic and industrial sector actors to develop or adapt imaging modalities. And as you know, France BioImaging is a part of Euro-BIoImaging and also of global bioimaging infrastructures. And among 8 regional nodes, we have 3 sites with strong expertise in plant imaging. The first one is very close to Paris, is Île-de-France Sud, and this node has a specialty about the observation of plant physiology and development. The Bordeaux one, which is mine node, and the topic of this node is concerning food development, plant pathology, and membrane lipids. And the last one, the Montpellier one, where they have an expertise for in situ and in vivo plant cell imaging. For example, in Montpellier, they developed a biosensor to visualize in live sample the redox state of iron, as you can see here in the root of Arabidopsis thaliana. They also develop a pH sensor to measure in different compartment cells the pH in live sample. And also they develop biosensor for simultaneously measure the chlorure and the nitrate and the pH. For Bordeaux, we have a strong expertise in the electron tomography, as you can see here. And we are very expert for plasmodesmata observation. We also implement a method for correlative microscopy. And here you can see, in fact, in light microscopy, a little structure which are tagged with GFP. And after we can focus on it to look the 3D infrastructure thanks to electron tomography. We also have a strong expertise in serial block space imaging. On here you can see some layers of cells of a root tip. And we also have a homemade system which allows to make a lattice-like microscopy. Also, we have a cell which allows to automatize cell treatment. And you have an example here with an automatization of the mapping of the cell-to-cell communication network. So now I finished, and I will give the screen to Clement for the Clem presentation.
So thanks a lot, Lisiane. So hello everyone. Today I will present you the relative light and electron microscopy approach that we developed here on Bordeaux Imaging Center on plant tissue. And I will focus on the work that I did during my thesis, which was about targeting ultrastructural events at the graft interface of Arabidopsis thaliana. So grafting is the act of cutting an aerial part of a plant, which is called the scion, that we combine to a rootstock of another plant. And as I just mentioned, the first step of grafting is actually cutting. And at this stage, all form of communication are interrupted, meaning long distance communication, and cell-to-cell communication through plasmodesmata. And as you can imagine, to make a viable graft, communication is really important and need to be reestablished. And this is exactly what we wanted to study with this very special focus on plasmodesmata. So the first technical issue with this project was to actually target where is graft interface. So here you can see a graft interface of Arabidopsis thaliana. And as you can see, all the cells are looking the same. And our first innovation was to actually use two different fluorescent markers, allowing us to identify the identity of each individual cell at the graft interface. Now, what is correlative microscopy and why? With fluorescence imaging, you all know that you can see interaction and dynamics because you can work with living samples, but you have a limited resolution and the not labeled structure are not visible. But here you can see on the image that it's really convenient to see the graft interface. With electron imaging, on the other hand, you have high resolution imaging and you have the cellular environment, but you need to work with non-living sample, fixed sample, and as you can see, there is no more fluorescence to find the region of interest. So correlative light and electron microscopy, CLEM, is consists in combining on the same sample an observation of the fluorescence and of the ultrastructure. And that way we can find back our region of interest, in our case, the graft interface. And more generally, you can use correlative microscopy for targeting events that can be rare or transient, for example. So we based our work on the in-resin fluorescence protocol that was originally developed by Kutsky in 2012, sorry, on yeast and mammals. And it's a protocol that is using high pressure freezing. They are able to do CLEM on the very same section. And we choose this protocol because they have a very high fluorescence preservation, antigenicity preservation, good ultrastructural preservation. And this protocol is compatible with electron tomography. And moreover, this protocol is compatible with non-adherent cell, which is our case with plant tissue. So here are the big guidelines we have for plant tissues. And first is to work with plants that have a strong signal, meaning a strong expression of your fluorescent protein. And of course, the choice of those fluorescent protein also appear to be something important. Then we reduced the exposure to chemical agent compared to the standard transmission electron microscopy protocol, where you have contrasting agents such as osmium tetroxide or glyceraldehyde. And to do so, we used cryo methods, and also we reduced the time of freeze substitution. We chose a resin that was polymerizing with UV, and to limit the photo damage due to the UV light, we reduced the length of polymerization to really its maximum to preserve our fluorescence protein. Also, we worked on the optimization of the thickness of the sections because usually with conventional electron microscopy, you have to work with a very thin section, about 60 nanometers, to get a good resolution. And you can understand that the thicker the section, more photon can be contained in the section. So we increase the section up to 150 nanometers. So this is a balance, because the more you increase the section, the lower the resolution will be with transmission electron microscopy, but I will get on that later. Also, optimizing the fluorescence visualization was really important. For example, the mounting, we have to really work on that to avoid the formation of air bubbles during the mounting. And plus, to acquire the full fluorescence of our sample, we had to perform Z-stack acquisition, followed by some projection to collect all the photon and visualize all the photon on the same image. So now I can show you some results on the graft of grafted hypocotyl of Arabidopsis. So you can see with confocal that we preserved the fluorescence. We had a good infrastructure, and we were able to combine the two to have the correlative view and find back the region of interest, our graft interface. At the graft interface, we were able, with transmission electron microscopy, to observe plasmodesmata. And because the sections were thicker compared to standard transmission electron microscopy, to improve the resolution, we implemented the electron tomography on those samples, which allow us to restore the resolution, plus to have the 3D information of our structure. And you can see on this cropped image that we can clearly see the plasma membrane, desmotubule, so the resolution we get in the end is really nice. So we were able to apply this technique on different projects and different issues. For example, I just present you with graft interface, which was on hypocotyl. We also worked with phase separation on hypocotyl, but with we also worked with root tip on 2 projects on autophagy and on plasmodesmata formation. And if you want to know more about correlative microscopy and set up your own experiment, you can find our paper. There is 2 papers, the plant physiology, which is the more biological paper, but reoriented on the technology, and you have the step-by-step protocol on bioprotocol. We have an online workshop on YouTube that you can find typing Clem Workshop BIC. And of course, you can come to visit us by applying to some fundings for your experiment through France BioImaging or Euro-BIoImaging. And now I want to thank all the people involved in this work from the lab, all the fundings, and I would like to thank you for your attention.